Hi, everybody. Thank you so much for joining us today. My name is Justin. I am a clinical researcher for Cure CMD, and I will be the session moderator today along with Gustavo. Hi, Gustavo.
Hey, families. Thanks for being here.
So today on this session, we hope to go into detail about the Dystroglycanopathy research. We are honored to have a couple panelists joining us today. We'll be discussing the projects that they've been working on. And hopefully we'll also have time to go through some of the questions that you as audience members have been asking us. So we hope to cover all those topics today. Now, but before we get this session started, there is a quick disclaimer that I must share with you. Let me go ahead and share my screen with you in just a second. And I'll quickly read out this disclaimer for us. So the information and opinions shared by our panelists will be generalized statements and are not intended to be interpreted as a promise for future treatment outcomes or a specific timeline for such outcomes. While we are all eager for advances, it is impossible for anyone to predict when a treatment may become available or anticipate roadblocks. The session is being recorded and will be shared on our public YouTube channel after the conference portal closes on August 31st. So now that we've gotten that out of the way, I would love to take a moment to have all of our panelists introduce ourselves. Why don't we go around and everyone can state their name, the institution that they're part of, and what their discipline is.
You want to call out an order?
Sure. Let's start with you.
My name is Dimah Saade. I am a pediatric neuromuscular neurologist, and I trained at the University of Iowa with Kathy Matthews and Steve Moore, and I did my pediatric neurology and neuromuscular fellowship there, and during those years got to participate in the yearly Wellstone Dystroglycanopathy family meeting. And I am currently at the NIH with Karsten Bonneman's group. And as part of that team, I focus on diagnosis for ultra-rare diseases, but also first-in-human therapies in children, such as the gene therapy trial for giant axonal neuropathy. And I'm stepping in today for Kathy Matthews, who could not make it today.
Thank you. Kevin?
Hi, my name is Kevin Wright. I'm an associate professor at the Vollum Institute at Oregon Health and Science University. And my background is a basic scientist studying nervous system development. And I've been working on dystroglycanopathy related research for about 10 years now, which started when I was a postdoc at Johns Hopkins and continued when I started my lab out here. And so for most of the work that we do in the lab, we use mouse models of dystroglycanopathy to try to understand how dystroglycan functions in nervous system development and function.
Thank you. And finally, we have Chiara.
Hi, everybody. I'm Chiara Manzini. I'm an associate professor at Rutgers Robert Wood Johnson Medical School and in the Child Health Institute of New Jersey. I'm a neurogeneticist, so I came to dystroglycanopathies now almost like 15 years ago, working on the genetics of different types of dystroglycanopathies, and my laboratory is still doing genetics, and we're working using zebrafish models to study the progression of the disease, but also to test possible therapeutic approaches. And I will tell you about it when we give our presentation. So thank you for being here.
Wonderful. Well, nice to meet you all. And on that note, why don't we go ahead and if anyone would like to start by sharing briefly what they've been working on in relation to dystroglycanopathy.
Should we keep the order, Dimah?
Sure. I was going to say, do you want to do basic science and then what is present in clinic? But I'll do just a clinical overview. Let me share my screen. And then let me know if I'm going a little bit over time. Can you see my slides? All right. And very briefly, I don't think this audience needs an introduction, but dystroglycanopathies are a group of autosomal recessive muscular dystrophies that are caused by decreased glycosylation or disrupted sugar groups on alpha dystroglycan, which connects the muscle membrane to the extracellular matrix. It is caused by mutations in more than 18 genes. And FKRP is by far the most common one. At the Iowa Wellstone Center, the Dystroglycanopathy natural history study aims at defining the natural history for the dystroglycanopathies. And as most of you know, the range of the disease can vary from severe muscle eye brain disease, congenital muscular dystrophies, all the way to limb-girdle muscular dystrophies. And the ultimate aim of that is to prepare for therapeutic clinical trials. And I have contact information down here for Carrie Stephan if you are not already part of that study. And as I said, most of the participants in that study have FKRP mutations, and this is the breakdown for the other mutations, the most common being GMPPB and POMT2. But as you see, a lot of the other mutations are underrepresented. And then just a breakdown of where patients come from in the US and then from Brazil and Canada. In terms of therapeutic options or research options for the dystroglycanopathies, most of what is in preparation or has been in clinical trial or is in clinical trial has been targeting FKRP because that is the most prevalent mutation. And so right now in development, as Karsten mentioned, there are many, I think more than 3 companies that are preparing for gene replacement, AAV-mediated gene replacement therapies for FKRP. And that should be coming into clinical trials in the next year or so. And then way down the line, in early phases, there are preclinical studies into genome editing and gene overexpression. There was one, I saw some mention of tamoxifen in mouse model, but that's also ways away. In clinical preparation and maybe starting soon this year is a trial of ribitol for FKRP. And then concurrently right now, there is one trial that is not recruiting out of Northwestern for weekly steroids and muscular dystrophies. And that trial has recruited patients with dystrophinopathies or Duchenne muscular dystrophy, as well as several other limb-girdle muscular dystrophies. Several studies have been closed or completed, but a lot of time that has happened either because of failure to recruit enough patients or failure to demonstrate or to meet efficacy endpoints. And so that was the case with the anti-myostatin and deflazacort, which is another form of steroid. And that highlights the importance of having a patient cohort and having defined natural history and outcome measures for any trial to be successful. So what that means for the other genotypes, and I'd like to go back to what Karsten said a little bit earlier. Really, knowledge learned from trials in other dystrophies is helpful, and improving, testing out the toolbox and fine-tuning the regulatory pathways, production, and testing it out in patients in clinic is extremely helpful and may open the way for a platform approach to gene replacement therapies. But that doesn't undermine the fact that knowing the natural history is still very important, and understanding the underlying pathological mechanism to identify treatment targets and limitations. And I put limitations here too. I think Carsten also alluded to, could the brain phenotype or brain abnormalities in dystroglycanopathies be tackled by gene replacement? And there's a big unknown, and there are limitations in that. And basically proving that one approach, be it ASOs, gene editing, or gene therapy, works for one of these diseases would help other diseases. And I took Kathy's cautious optimism illustration. Technically, the glass is full of air and water. And then I'll end with another save the date for the Dystroglycanopathy meeting that's upcoming in July. If you haven't registered, please reach out to Carrie, Stefan. I think the goal mostly for the coming few years would be to recruit more non-FKRP dystroglycanopathy patients and engage them into what are outcome measures that would be meaningful for them, what are targets of improvements that would be meaningful for them, and also trying to identify similarities in those severe dystroglycanopathies across the different genetic causes and genotypes. So with that, I will end and pass it over to the next person.
Thank you, Dimah. Kevin?
Okay.
Sorry, Kevin, before you start, I want to stress again that for the people watching, we want you to participate. If you have questions for any of the panelists, go ahead, type it there in the chat. Or unmute yourself and ask. Thank you. Sorry, Kevin, go ahead.
Thank you, Gustavo.
Okay, can you see my screen here?
Yes.
Okay, wonderful. Okay.
Let me introduce you from the QCMD. Kevin Wright is our grantee. Thank you to the families that fundraise and support research in alpha dystroglycanopathies for this grant cycle 2020 to 2022. We selected with our scientific advisory panel, we selected this work to be funded for the next couple years. So yeah, go ahead.
Thanks, and I really appreciate the support from CureCMD. So I decided to just show two really basic slides just to kind of highlight sort of the direction that our lab is going with this project. So we use mouse models to try to study sort of the basic function of dystroglycan and nervous system development. And previous work from my lab and many other labs has focused a lot on the role of dystroglycan in early brain development, where it's required for sort of acting to maintain these cells called neuroepithelial cells that sort of act as a scaffold for how the rest of the neurons migrate in the brain and form axon tracts to connect different parts of the nervous system. But more recently, my lab has started to focus on what dystroglycan is actually doing later in nervous system development. And we've known for a while that dystroglycan is present on neurons themselves. And so in order to be able to sort of isolate what dystroglycan is doing on neurons, we sort of use these genetic tricks in mice where we can use a mouse model that has basically the ability for us to delete dystroglycan selectively from different cell types or at different times during development. And so just in these upper panels here, I'm just showing sort of the mouse model that we use, where we use this conditional knockout mouse and we cross it to another mouse that will allow us to basically delete it selectively just within pyramidal neurons. And so this is just confirmation that we've deleted dystroglycan. So normally we see, if we use an antibody to stain and that lets us look at where dystroglycan is actually located, we can see that it's on these neurons here and that disappears in our knockout mice. And so One of the major things that we found, and this work is actually in a paper that we just had accepted for publication, is that when we delete dystroglycan from neurons themselves, we actually lose an entire class of their synaptic partners. So synapses are basically how neurons talk to one another. There's lots of different types of neurons in the brain, and we're focusing here particularly on the cortex and the hippocampus here. And so we can use antibodies to basically identify these different types of neurons and sort of look at where they're at in the brain and how they're connected to one another. And what we found is that if we remove dystroglycan selectively from these pyramidal neurons, we actually lose an entire class of their presynaptic interneuron partners. And these are neurons that are marked by their expression of a protein called CB1 receptor. This is the cannabinoid receptor. And so normally, if you look at sort of a side view of a mouse brain, you can see that there's this nice staining that labels these CB1 receptor positive neurons. That's all throughout the olfactory bulb in the mouse, the cortex, and the hippocampus. And that's completely gone when dystroglycan is deleted. And so in the second slide, I just have sort of a more zoomed in version here, focused specifically on the hippocampus. And just as a comparison, for comparison's sake, showing a different type of interneuron marked by a different antibody here looks completely normal in these mice where we've deleted dystroglycan. So these are the controls. These are the ones without dystroglycan. So that all looks totally normal. But when we look at the CB1 receptor positive interneurons, we can see that instead of forming these nice synapses in the hippocampus, that these neurons are completely gone. And so work in the lab right now is focused on a couple of different questions sort of related to this. The first is we're trying to understand what happens to these neurons. We think that they die early in development. And it's really important for us to figure out the timing of when this happens, so that we know if we want to try a therapeutic intervention, we have sort of a window as to when we can actually try to rescue these neurons. The other question is whether other neuron subtypes are affected in other parts of the brain. And so we have a couple people in the lab working on looking at the role of dystroglycan in the cerebellum to see if it plays a similar role in maintaining synapses between cerebellar neurons. And then finally, one of the things that we're really interested in is testing the possibility of using a gene therapy based approach to see if we can rescue this phenotype. And so we're developing AAV-based approaches right now, where we can come in and restore dystroglycan expression very early in development and ask whether or not we can actually get these neurons to hang around if we can do that. And of course, finally, we're also testing this in sort of models that sort of more accurately represent human patients. So we've collected a couple of mouse models of dystroglycanopathy. So we have mice with mutations in B4GNT1, FKRP, and POMT2. And we're in the process of analyzing those mice right now to see if these same types of neurons are affected in those models, and then whether we can actually rescue those neurons by restoring the respective genes in those mouse models.
Great, thank you, Kevin. Hi, Francesco. Thanks for joining. Do you want to introduce? Can you introduce yourself just briefly?
Oh, yes, my name is Francesco Muntoni. I'm sorry I'm late because I'm coming from a clinical meeting, and I am a pediatric neurologist. I work at the Institute of Child Health at University College London at the Thomas Hospital. Thank you for inviting me.
Thank you for being here. So now, Chiara, you want to show us what you are working on?
Sure.
Thank you.
Let me share my screen. Oh. Is it all dark? Wait, let me try. I know. Can you wait a second, because it's doing something funny when I have my.
Yeah, we see both.
Yeah, the presenter. It's like when I do the. Sorry. Okay, now I can get it. I have two screens and it's messing with my share screen. Okay. Now it should be okay. So I just wanted to give everyone and the families a background of what my lab is doing. So we've been working on dystrophy genetics for a long time. And here I'm just showing kind of like the progression of how we started from like very large families and needed multiple affected children. And now we can move a lot faster, and kind of the genes we've been involved into either identifying or doing mutation analysis. And I think one of the questions that popped up in the pre-questions that we got was, why aren't we finding anything else? And why are there so many individuals that are still not diagnosed? And one of the focuses on my lab right now is actually to trying to find non-coding mutations. And so there's a possibility that some of the kids that still don't have a genetic cause may have a mutation that we can't identify with the regular exome sequencing approaches. And so we have a study right now focusing on that. And so if anybody's interested, I would be more than happy to talk to families and get started on some of those projects. Let me see. So the other thing, as I was telling you at the beginning, we work on the zebrafish, and I think Karsten in his beautiful presentation mentioned a little bit about zebrafish and why to work on a fish to study a human disease. And so I wanted to give you a little bit of background on that. I mean, one of the reasons is that it's a vertebrate. It has a pretty good complexity related to just doing work on a cell culture. We can look at the muscle, we can look at the brain, we can look at the eyes, we can look at development. Has great statistical power. There are many embryos that are generated from very small number of mothers that we can follow. They develop really quickly so that we can get a phenotype. Like the one that you see at the bottom with the little fish having like issues with its tail, its size, and its brain by 3 days after fertilization. It's transparent embryo, which allows us to look at the tissue. It's a fully sequenced genome with very good functional homology to humans. And so that was one of the reasons I actually my lab also does mouse like Kevin's on other chemist group and on other projects, but we've really focused on the zebrafish for Dystroglycanopathy, just of this sheer power to be able to model multiple mutations because it's such a heterogeneous set of conditions, and to try and find possible therapeutic approaches. And so we have developed, as you see the little fishes, we have developed multiple fish models, either with knockdown or with mutants. Right now, the ones marked with R are the ones that we've developed at Rutgers. My lab just moved to Rutgers a couple of years ago to be able to develop more of these models. And so right now we're working on POMT1, POMGNT1, FKRP, and POMGNT2 models, and we're developing more, and we're validating some of these zebrafish models to be able to, the first step, I think, is going to go after the low-hanging fruits. And so to compounds that are currently in development for other muscular dystrophies and for other muscle disorders, and see if they can be effective. I mean, of course, like the ribitol treatment and some of the approaches that Dimah talked about earlier, to see if there's anything that can be translated as quickly as we can, as all of these approaches with AAV and gene therapy come into play. And so these are some of the things, and we're also very interested, of course, in looking at the balance. So I'm a neuroscientist by training. I particularly care about the brain. So really looking at how all of these treatments will affect both the muscle and the brain at the same time, because you can rescue mobility, but because it's a very early developmental defect, to what extent can we push the synapses that are already present in the tissue to function better, to improve function. And I think this is a concern of many of the many of the parents also getting improved cognitive function and communication and vision for these kids. And so I just wanted to do a shout out to our Zebrafish team, which is Dr. Brittany Karras, like to Tex, Brian, and Kristen, and several students, and our funding from the National Institute of Neurological Disorders and Stroke. This is my contact information. If you want to reach out, we'd be happy to chat.
Thank you, Chiara. I would like to, Francesco, I don't want to put you in the spot, but we were talking about in the plenary session that Karsten mentioned about the specific case of the dystroglycanopathies, that the muscle and the brain are affected. And for the gene therapy, for example, that you have to address both at the same time. Is that possible? Is that possible using the same vector? Should you use two different vectors? I don't know, any comment that you can make or want to make?
Well,
very difficult question, I know.
Thank you, Gustavo, for this. Well, I suppose there is a pros and cons for many different things. You could see that, for example, a small molecule approach will be probably have a easier way to, if you deal with an enzymatic defect. I'll make an example. A small molecule approach will have easier access to multiple tissue. I think in terms of the AV gene therapy, I think there are three barriers, I think. One is the AV that you use and the biodistribution of that AV. The second is the promoter, because if you really want to recapitulate brain and muscle, I think you almost obliged to go back to the promoter of the gene you're interested, because these are not necessarily ubiquitously expressed protein. So if you want to be started to become very precise with your question, then it needs to be a precise promoter to drive that. And the third, I suppose, is a bit like Chiara just mentioned, and also the colleagues earlier on, how much of this is developmental. You want postnatally be able to repair the pia-glia limitants if that have been during development affected. But there may be many aspects related to neuronal function. And I think I was interested about these different type of receptors that have been now more recently identified. But, for example, GABA receptor has been heavily involved in dystroglycan function. And there is perhaps some hope that restoring dystroglycan and potentially dystrophin may be relevant as well. So it's a very long answer to say that I think there is hope, but I think that a little more work underpinning these hopes is necessary. That is why I think, if you like, the work QCMD is funding to try to address this and make sure that expectations are reasonable, and but also some of these technical bottlenecks. What is the promoter of FKRP and whatever. It's easy to say, but to clone it, to put in a viral vector to be delivered to a mouse, well, nobody has ever done it yet.
Thank you. Hi, Kevin Campbell. We have two Kevins, Kevin Wright and Kevin Campbell. One has a little bit more experience, just a little bit more experience in the area. And we cannot hear you, Kevin Campbell.
I'm sorry, I didn't receive the password. Remember when I registered before, but now I'm on. Okay.
Okay. Can you introduce yourself?
Oh, my name is Kevin Campbell. I direct the Wellstone Center at the University of Iowa. And the center is focused on the dystroglycanopathies, trying to understand the pathogenesis and to develop the clinical techniques to go on for clinical trials.
Thank you, Kevin. I don't know if you, Justin, we are having some questions or some of the ones that were already submitted, if we can discuss that. We don't have too much time. I think 8 more minutes, right?
Yeah, absolutely. I think the majority of these questions have already been answered by our clinicians and researchers, but I guess one closing question really that our audience members are dying to know is, do you think we will continue to identify genes that lead to a form of dystroglycanopathy? Because there are still so many without genetic confirmation, but all of the characteristics.
I would say yes.
Kevin, Chiara, yes.
So we have a neuropath conference that meets once a month, and we started this a long time ago. And every year, probably in the last 10 years, I think that this is the last year for identifying new genes, and there's new patients come along. I think probably the next group of dystroglycanopathy genes might not be as selective for just dystroglycan. So some of the trafficking genes might, also affect other glycosylation pathways, but including dystroglycan. So I think that, the patients might have additional symptoms, or different symptoms because they might be affecting more than one pathway. But I think that it has, I would guess there's probably going to be another 5 or 6 genes if I had to put money on it. Francesco or anybody else?
Well, well, Chiara has just given a presentation, and we are working on one possible new gene, but we still need to be sufficiently certain that then we can even email you and Chiara and so on without making an embarrassment of ourselves. And I suppose in the past you published the complementation technique that was also very elegant, and especially when it comes to potentially intronic mutation or regulatory mutation as opposed to new genes, that was also a very powerful technique. So I didn't discover that, you discovered that.
No, yeah, but one problem with that technique is that the genes like the mild FKRP mutation, if you look at skin fibroblasts, they are completely normal. And so I think there's going to be genes that in the fibroblast state will not really show a defect. Muscle and the CNS are much more complicated than the fibroblasts in terms of trafficking and what needs to be done. And so I think that that's going to be hard to use that technique. Toby Will actually tried to infect them with MyoD to make it into myoblast and then see, but that didn't work too well. But I think that clearly that technique will pick up the severe cases. We find that even if we knock out genes in HAP cells, the ones that are mild, that don't really give a phenotype in the HAP cell in culture. So I think the cells in culture, probably can compensate for the small amount of defect versus, complex tissues like muscle or neurons or, astrocytes, cannot.
Thank you very much. We have a few minutes before we have to bring this session to a close. So I'd like to finish this up by having our panelists have a take-home message for our audience members. Why don't we start with Dimah?
I get the easy task. I guess the biggest take-home is it is important for patients with ultra-rare conditions to be followed consistently and join natural history studies to better delineate the natural history of the disease, and also know what are outcome measures or what are improvements that would be valuable for families while there's a number of promising therapies coming through.
Chiara.
I just saw there's a question, and actually I was kind of going to address that. I mean, it's like the message is that I think there's hope. As somebody coming from the neuroscience field where in neurology we have cured very few things. I mean, we just started curing things, and having looked at the enormous success of the neuromuscular disease field for other conditions, I'm really hopeful that we will be able to fix the muscle. And the question is like, how are we going to be able to fix the brain? Because this is a neurodevelopmental condition. And working on cognitive deficits also that have no neuromuscular component, I'm really hopeful that we will be able to push the brain to at least regain some function. And the disclaimer is that because this is early developmental and the intervention has to be at birth, you're never going to get a normal looking brain. But I'm still hopeful that with the studies that we're doing, that Kevin Wright is doing, looking at synaptic function, that will be something that will be possible. I don't know how soon.
Yes, and that was the discussion when we received when we were evaluating the grants proposals, and we know how hard it would be to fix the brain, but at least with the work like the one that's done in Kevin Wright's lab, to try to see if there is any possibility to rescue at least part of the synaptic plasticity and have some good outcome, at least to improve at least a little the quality of life in the cognitive impairment area. That's something that families have asked us. They are worried about that. They talk about that all the time, most than the muscle aspect of the disease. So with at least in little steps, we want to get there.
Kevin?
I would agree. Steve Moore at our neuropath meeting sometimes shows a patient's muscle biopsy, which looks really severe. And then Steve says, and this patient's still walking. So sometimes there's a disconnect between the pathology and the function. The problem with the muscle biopsy is you're only looking at one muscle at one point in time. So you're not looking at all the muscles. But I think the same thing is true for the brain. I would, think that you might not be able to repair the developmental aspects, but maybe you might be able to repair the synaptic function, and, I think we have the tools. I think Kevin Wright can do this. There's been some reports on genes that cause abnormalities in LTP. And, you clearly could now design, systems where you put in vectors later on and see if you could restore LTP. And so I think there's the tools to try to address that. The one reason I'm optimistic about it is I've been getting contacted by people that are also interested in the cognitive defects in DMD boys. One of my former chair of physiology here at Iowa, his grandson has DMD, and their biggest problem is the CNS abnormalities. He's a milder muscle, in terms of muscle problems. So I think, if we team up with the DMD community for the CNS studies, I think that'll give us, this much bigger patient base. And there might be some common, things that could be addressed between DMD and the dystroglycanopathies. And so I think that would help.
Kevin Wright.
I'm also hoping that, some of the, the advancements in fixing brains could be applied to like older scientists. Like myself.
If anybody else wants to give any thought, Francesco,
yeah, I can just chime in really quickly too, and sort of going back to the question, as Chiara touched on, there's not going to be much that I think we can do with some of the really, really early developmental defects that arise. But one thing that is sort of nice about the mouse models that we have is that we're going to be able to test what happens when we rescue these things after birth in that context of either a mild case or a severe case. So having this sort of whole range of animal models that sort of have the full range of clinical presentations, if you will, is a really powerful tool for us to be able to test some of these therapies. And I'll also say as a plug for basic science research, dystroglycan is a tricky molecule to work with in the sense that it does so much. and we're constantly learning new things about what it's doing in the nervous system. And so I think that, you know, what's nice is that we have some really good genetic tools to be able to sort of dissect these different roles for dystroglycan apart during development, and that'll help us better understand what we actually need to be doing in order to correct some of the defects.
I think very briefly, I think I like the work that was mentioned regarding the Duchenne muscular dystrophy. I think in Duchenne muscular dystrophy, coming back to the point of GABA, the postnatal restoration of dystrophin relocalize GABA at the postsynaptic density. It improves behavior. So I think that there is some hope. And I think while it is frustrating probably for ultra-rare disease not to have an immediate treatment, the good news is that there is a lot happening in terms of biodistribution of these different vectors in other conditions. And the field is learning, is moving at a pace that is really unprecedented between either delivery of viral vector to the central nervous system in spinal muscular atrophy and to muscle in Duchenne muscular dystrophy. And little by little, the amount of information is really exponentially increasing. So I think that I think some of these may well be amenable to improvement postnatally.
Well, thank you to all of our panelists and our audience members for joining us today. If anyone would like to continue the conversation, I would like to direct you all to our community lounges, which you can find in the SciFam agenda. And yes, we hope that you enjoy the rest of your SciFam experience, and have a good day.
Yes, and we have a lounge for families if you want to hang out there and find some other families to talk with. And thank you everyone. Chiara, Dimah, Kevin, and Francesco. Thank you so much.
So this is our first day at Iowa where we don't have to wear a mask.
Oh, congratulations.
Everyone is free, so it's exciting.
Thank you, everybody.
Bye.
Bye.